Hi, everyone. This is Caroline with Fiveable. Thanks for tuning in tonight. I'm glad we have a decent-sized crowd. So obviously, here we are. It's Wednesday evening. If you haven't joined us before, we are going to be jumping into review tonight. So this is review part 2. So last week, if you weren't able to attend, we talked a lot about evolution, natural selection, the different ways that alleles can change and are modified. Then we moved a little bit into macroevolution, which is where we're going to pick up today. So we'll kind of tie a bow on evolution. We'll talk about macro. We'll talk about speciation a little bit. And then we're going to jump in to talk more about cells, cellular transport, and then cellular energy. So we're going to talk about cellular respiration and photosynthesis. Now, I will be the first to admit that this is an incredibly ambitious plan for a 1-hour session to go through the rest of evolution, cells, cellular transport, cellular respiration, and photosynthesis. And if it doesn't happen, it doesn't happen. We can push things back to next week. We do have a little bit of freedom, but that's the goal. So we'll try to get through everything. That being said, if you have questions at any point, please ask them, because that's what's most important, right? Like, I'm more interested in answering your specific questions than reviewing every ounce of material, because there's so much to review. And if there are specific things that you are struggling with, I'd rather be able to focus on them with you so that you feel comfortable and confident, because exam time is just around the corner, as I'm sure you know, as I'm sure you've been feeling. Obviously, if you want to watch replays of videos all the way through back in September, there's probably 30 videos, I want to say, give or take. You just need a Fiveable Plus membership, so you can register for one of those. That will also give you eligibility into some prime kind of cram time sessions right before the exam. Okay, I think those are all of my logistical things. Let me think. No, that should be it. So we're going to jump back in. And again, if you weren't with us last week, we're kind of jumping in. You're going to be in the middle of evolution with us. So bear with me. Okay, so picking back up to speciation. So when we left off last week, we were looking at this image, which is depicting the difference between allopatric and sympatric speciation. So allopatric truly means other land or other country, and this is speciation that takes place when there's some sort of geographic barrier between populations. So something to be very familiar with is that in order for speciation to occur, right, in order for one population to all of a sudden become two different species, there needs to be a halt in the gene flow between those two populations. Because if they keep on reproducing, that's like normal life, right? That's a normal population, and they're going to continue to be the same species. But if speciation were to occur, that would mean that there would need to be a stop to that gene flow so that they're no longer reproducing and they become reproductively isolated, which is in and of itself what creates, or what we define as different species. So allopatric, this occurs with a geographic barrier. Usually allopatric speciation is a little bit more common just because, as you can see, there's an actual physical barrier in place to prevent gene flow between these two populations. So therefore, they're evolving separately. Perhaps their environments have slightly different habitats, different predators, etc. They're more able to evolve in their own path, and they're also not seeing one another, and therefore mixing their gene pools. On the right-hand side, sympatric speciation is going to be a little less common. Sympatric means same land or same country, and so these are happening without a geographic barrier. Still totally possible, just less likely because they're bumping into one another all the time. So I always like to, tell my students, it's like if you break up with someone and you move away, it's way easier, right, to stay apart and not get back together than if you break up with someone and you see them every day, right? It's like the difference between allopatric and sympatric speciation, right? Sympatric, they're seeing each other every day, so it's a little less common that they're going to be able to successfully prevent gene flow. Anusha, I would love to define gene flow. So gene flow is exactly what it sounds like. It is the flow of genes. So it's usually something that increases genetic variation. And like an example would be if you move to a new area and you start a family there, right? You are now contributing your individual gene pool, which is the genes that you possess, into this new population. right? So it's just the movement of genes or alleles between organisms or populations. So as you can see on the left-hand side, there's not going to be any flow of genes between the bluefish population and the orangefish population because of that geographic barrier. They're not going to be reproducing, right? Which is when genes would flow between groups, opposed to on the right, where that would be more common and more possible. So hopefully that helps there. I may have a picture of it from last week. I'll have to take a look if you still want further explanation. Okay. Okay, so some examples of these things, because that always helps make it easier. Just like another picture that shows this allopatric speciation. We have this crevice that's running through this population of trees, and we can see that that's leading to two separate evergreens on either side. And again, it's less likely that pine cones and pollen are going to fertilize the trees that are across that barrier. So they're more likely to just be reproducing on their side of the barrier. And on the right-hand side in Sympatric, we have a new population sprouting up in the middle there, which is again going to be a little less common, just because all of those trees are capable of pollinating one another. Okay. Another example of allopatric speciation right on either side of the Panama Canal are these two now separate species of porkfish that used to be the same species, but then the Panama Canal got built, they got separated, and now you can see that they've evolved into two separate species with a very recent common ancestor. It's just a real-world example for you. An example of sympatric speciation that's really common, and if you've ever watched Bozeman's video on speciation, he refers to this. It's with apple maggot flies. And basically, these apple maggot flies started to tend to have preferences for either apples or hawthorns, which are more like crab apples, if you've heard that terminology. So you can see in this population of trees right here, I should get my pointer out. We have a mix of crabapple or hawthorn and apple trees. And as these maggot flies decided to kind of tend towards one versus the other, it actually changed the way that they smelled to potential mates. And so a mate wanted to pick another mate that ate the same food as them. So the apple flies, maggot flies, mated with other apple maggot flies, and the hawthorn maggot flies mated with other hawthorn maggot flies, and they actually arose into two separate species, even though they're living in the exact same forest. So that would be an example of sympatric speciation. There's still some sort of barrier in place. In this case, it is the location of the food and the food itself and how that presents itself, but they're still living in the same area. So hopefully that provides a little bit of clarity. Okay, Alan, you're asking a great question. So here's my my long/short answer. So it says, do these reviews cover most of what I'll need to know on the AP test, or is it general review? So the problem is this: I will go through as much as I possibly can, but I only have an hour once a week, right? And when your teacher probably taught evolution, like when I teach evolution, it's like every day for 5 weeks, right? And I have like 75-minute class periods. So if you think about the amount of time that I have to cover topics, I'm certainly not going to be able to cover everything that you need to know on the test, but I can at least give you an overview of the most important things. And sometimes I feel like what I'll tell my students to do with a session like this or looking through a review book is just write down the things that you don't know and get more information on those topics. So if you feel really great about speciation, then you can write that in your, yep, I feel super confident about this list, and kind of move on. But if you don't, you can add it to your, ooh, I need to either ask Caroline for more help there, or ask a clarifying question during our Fiveable review, or look it up or watch a Bozeman video, look it back up in my textbook. Because if I covered everything in the textbook, I mean, imagine how long it would take to read. Your textbook's probably like 1,200 pages, which is nuts. Do you need to know all the vocab in your textbook? Absolutely not. So the good thing, and I'll try to incorporate this as frequently as possible. The good thing about the redesign of the AP test, which happened in 2013, is that it's a lot less vocab heavy and a lot more application based, which is why I'm trying to cursory go over these words, because you do need to be familiar with them. But more importantly, I'm concerned with you understanding what they mean and applying them to new situations, which is why I'm giving you these examples like sympatric and allopatric speciation. Because if you look at old AP exams, or if you look at old free responses, they're going to throw new topics at you, and it's not really necessary that you know all the vocab to define it. You just need to be able to think critically and apply some really good bio knowledge to those scenarios. So I can't tell you that just listening to these is going to totally prepare you for the AP exam, because it's not nearly enough time for that. But it is a good jumping point to tell you what you do and don't know, and a good opportunity to ask some questions if you have specific ones. Hopefully that helps a little bit. And I will try my best to obviously cover as much as I possibly can. Okay, so going back to decreasing gene flow. So sympatric speciation, again, is obviously more complicated because they're living in the same area. So a couple ways that this can happen: habitat differentiation, sexual selection, and then polyploidy, which is really common in plants. It's a good one to know about. It's one of those illustrative examples that teachers like to talk about. So this example with the maggot flies is actually kind of a dual example of habitat differentiation and sexual selection. The sexual selection part comes in because the smell of the fly is different based off of what they're eating, so that's helping them pick a sexual partner. But it's also habitat differentiation because while they're all living in the same area, they are kind of manifesting on different trees, which gives them a slight, like they're not going to be bumping into an apple fly if they're on a hawthorn tree as readily. So it's kind of a mix of those two examples, but those are good things to put in your back pocket to know of that would decrease gene flow in a sympatric speciation example. If you were asked to write about a scenario in which this speciation might be more prevalent, any of these three would be good. And I'll talk about polyploidy in a second. So habitat differentiation, another example of this, if you've ever done the HHMI lizard phylogeny situation, if you haven't, it's really cool. But basically, there's a bunch of lizards that live in the Caribbean, and they used to all have a common ancestor. And then they differentiated based on their habitat. So some live on the ground, some live on trunks, some live up in the leaves. And you can tell based on their body types that they have adapted to those areas. They all have different lengths of legs depending on if they're grabbing branches or running on the ground or what it may be, but they've all kind of differentiated themselves on the same tree, just in different ranges of it, and that's where they reproduce. So that's one way that sympatric speciation and the decrease in gene flow can occur based on living in and navigating different areas of the same habitat. With sexual selection over time, and this is a very simplified example with dinosaurs, how exciting, and they're picking just dinosaurs of the same color. But oftentimes this will stem from like a certain pattern. If it's birds, they may be looking for a certain pattern in a sexual partner, and that can lead to speciation over time. Often it's like smells again, or pheromones that are given off by insects based on what they're eating. So if they have different eating preferences, then they're going to be mating with different partners, but anything where they're kind of differentiating themselves based off of some sort of characteristic would be an example of sexual selection. And again, if they're only reproducing with that certain individual, then that could lead to speciation because it is truncating that gene flow in between populations. And then lastly, polyploidy. So polyploidy is very common in plants. And what happens is, just to reframe, because you definitely need to know these vocab: haploid, diploid. Okay, haploid is like half the amount of chromosomes. Haploid would be like the number of chromosomes in our sex cells. So in egg and sperm, we have 23 chromosomes, one of each chromosome number 1 through 22, and then either an X or a Y. Diploid is all of our body cells, right? So we have 2N or 46 chromosomes because we have two versions of each chromosome 1 through 22, and then you either have two X's or an X and a Y. So polyploidy means you have more than that. And you can see two examples here. There's triploid and tetraploid. And basically it's just an error in mitosis, which we're going to talk about next week. If you feel a little bit uncomfortable with some of these terminologies, next week is when we're really going to dive into DNA and alleles and more genetics stuff, as well as mitosis and meiosis. And I've definitely sang a song on here before about those, but I'm not going to give it away now, so tune in next week. But basically what happens frequently in plants, it happens in humans too, but oftentimes the offspring doesn't survive, is that the gametes just never separate themselves. And so when fertilization occurs, usually self-fertilization occurs, there's way too many chromosomes. So you can see the parent here is just a diploid with 6 chromosomes, and the offspring is a tetraploid with 12 chromosomes. And what this does is this plant now can only reproduce with itself, which plants do. Plants are asexual and sexual beings. But this plant now can only reproduce with itself, okay? And so it will produce offspring. And then those offspring, once they grow and develop, can start reproducing with other tetraploids in the area. But that's a really easy way to lead to speciation, because the tetraploid and a diploid plant would not be able to crossbreed, would not be able to cross-pollinate, and therefore they have effectively created a new species in just one generation, which is crazy. But that's kind of an important one to know about. So definitely put that in your back pocket as well. But again, it's really just a non-disjunction in meiosis that leads to too many chromosomes in a plant. And therefore, when it has too many chromosomes, it can self-fertilize and therefore kind of make its own population, but it's not able to fertilize with any of the parent plants. Okay. So this can all lead to adaptive radiation, which is basically going to be where we have a vast diversity of, usually vast diversity of environments, because environmental pressure is going to be our main reason to evolve, right? Is not having enough food or having some sort of problem or crux in the environment that's leading to this push of natural selection. So we can see stuff like this, like with the Galapagos finches, which are obviously the most common example of natural selection. But because of the diversified food sources on all the different islands, we just saw this boom of natural selection occurring because of the vast environmental pressures on each of those. So there's a vocab term for you, but truly it just means that we're having this kind of like radiation, this radiant, think of like a sun's rays coming out here from an ancestor, usually based on diverse environmental pressures. Okay. So phylogenetic trees, I'm just going to touch on these briefly. For whatever reason, students seem to hate these. I don't know why. I'm going to show you one of those tables, too, that you can use to construct a phylogenetic tree. But I will also say to you this. I can probably guarantee that you will have to analyze a phylogenetic tree in a multiple choice question. But that should be a relief to you, because that's easy, and I can show you that it's easy. What I feel pretty confident in saying is that you probably won't have to analyze or construct a phylogenetic tree in a free response question. Sorry, everyone. Because it was the first long free response question on last year's AP exam. And usually they diversify. Like if they really hit phylogenetic trees in a big way, which they did last year, then you wouldn't be responsible for covering something in that magnitude. So maybe that gives you some peace, maybe not, I don't know. But that's my insider's opinion as far as the fact that you probably will not. I would be very surprised if you had to construct a phylogenetic tree or analyze one in a free response, but multiple choice for sure. So all phylogenetic trees tell us is what the most recent common ancestor was between two species. So as we're moving down here, the top of the screen is going to be the very most recent common ancestors. So right off the bat, I can tell that these two on the left, looks like a rat perhaps and a rooster, have the most recent common ancestor of all the species listed here. And I know that because their most recent common ancestor is the furthest to the top of this tree as any other, right? And these lines are representative of time, okay? So the next most recent common ancestor would probably be a tie between this rooster-rat pair and this frog over here, as well as these two fish over here. Because as you can see, if this is our time continuum, they're going to line up about the same time as far as common ancestors go. So if you were analyzing something like this and they were posing a couple question options to you, you would be able to decide how closely related something was based on how recent a common ancestor was. Another thing to remember, right, so let's say, okay, all of a sudden we get tetrapods, right? We have our four-legged individuals, okay? That's evolving right here. That means everything after this point should have four legs or some version of it, right? Because like We have hands, roosters have wings, etc. Okay, but so it's important to remember because they might ask you a question of when did blank evolve, and you'll want to find that subset of organisms that have that trait, and then trace back to what their most recent common ancestor is. In order to see when that may have evolved. And oftentimes they'll include a timeline over here so that you can actually pinpoint specific times of common ancestors or when things evolved based off of that. Something else that's really, really important that I always try to talk about with phylogenetic trees is that organisms that exist today did not evolve from one another. So I can give you the most common example. Humans did not evolve from monkeys. Like you can't say like a human came from a gorilla or a chimpanzee. Not true, because if we did, then the chimpanzee and the gorilla would no longer be living because we would have, evolved from them, right? So what is true is that humans and chimpanzees have a very recent common ancestor, our most recent common ancestor. We are most closely related to chimpanzees. We have like 99% the same DNA as them. Okay, so very recent common ancestor doesn't mean we evolved from them, right? Because if we did, they wouldn't exist anymore. They would just be a little junction right here on the tree of life, okay? But instead, here's our common ancestor. Here's us. We're not a rat, but here's us in this fake example. And here's the chimpanzee. And again, most recent common ancestor did not evolve from them. So yeah, never write that on an AP exam because you'll make some bio teachers angry. But I'm sure your teacher has talked to you about that. Okay, so this is common. And again, I don't imagine that you would have to construct one, but they may give you a table and you may need to analyze like who would belong on a sister taxon, or who is the most closely related or least closely related to who, based on one of these tables. And something that my students always say is that they're really, really bothered that half the table is empty. Okay, so what I always tell them is these empty cells mean nothing. It doesn't mean, right, that E. ferris is not closely related to anyone, right, because this whole column is blank. It just means that that data lives elsewhere. So these tables are redundant. They have the same column titles as they do row titles. And therefore, if you want to figure out how closely related E. ferris is to everyone else, you need to look at the row for E. ferris. Okay, and whoop, there all the numbers are that you were looking for. So if you ever see a blank cell, like if you're looking for E. ferris and G. gallus gallus, it doesn't exist here, it exists elsewhere. So here we go, E. ferris, G. gallus gallus. There it is, 11 differences. Okay, so again, the blank cells mean nothing. They just mean that information lives elsewhere. You can always copy the numbers over if it really bothers you, but I think that's a waste of time. So when you're attacking one of these problems, the first thing you want to do is look at the title. Always important. But this one says the number of amino acid differences in cytochrome C among 5 species. Something that I would get used to is when you see words like this, I don't want you to panic and be like, I don't know what cytochrome C is, because it truly doesn't matter. So you can even skip this word in your head if you're like trying to sound out, and cytochrome is not a terrible word, but sometimes they put terrible words in there. They usually don't matter. So you can say the number of amino acid differences in 5 species, and that means the same thing, right? We're just looking at how closely related things are, right? So these tables will either show similarities or differences, and the title is very important in deciphering between the two. This one is differences, so lower number equals more closely related. So right off the bat, I know that I'm looking for low numbers to show who's closely related. I see that E. africanus and E. ferris only have one difference. They're very closely related. They need to go together on a sister taxon. A sister taxon would be like this little V right here, okay? Like your sister, you're closely related to your sister. Okay, I'm also noticing another low number between G. gallus and A. forsteri. So I know that those two are also really closely related, and they need to go on a sister taxon as well. Now, without even looking any further, I can then conjecture that D. polylepis is going to be our outlier here on this outgroup arm by itself, but I can also clarify and see that D. polylepis has 18, 17, 20, and 21 differences with everybody else. So they are very, very far away and not very closely related to any of these species. Okay, so I would put D. polylepis all the way to the left. I would make sure that I had A. forsteri and G. gallus together, and I would make sure that I had E. africanus and E. ferus together. Now, the cool thing is, and you can get super technical and try to figure out which pair is more closely related to D. polylepis than the other, but it really doesn't matter, and you can tell that it would be G. gallus and A. forsteri because they have less differences than the E pair, okay? But it truly doesn't matter because anytime you have an intersection like this, you can rotate around that point, okay, without changing the meaning of the phylogenetic tree or the cladogram. So I can rotate around that point, meaning it doesn't matter if I put the A. forsteri and G. gallus pair here or here, because I can rotate around this point and it won't change anything, okay? It doesn't matter in the same vein if I put G. gallus here or G. gallus here, because I can rotate around this point and it'll still mean the exact same thing. So there was a question on the first long free response of last year's AP exam that asked, they showed a phylogenetic tree and they said, which two species could you switch without changing the meaning of that tree and those relationships? And you had to look for where we had like a most recent common ancestor and just rotate around one of those points without changing anything else. So it does come up on occasion. And there's going to be your answer, which I just showed you guys. But again, not super complicated, as long as you're reading closely and you know if you're looking at similarities or differences, you should be able to kind of not get too overly concerned here. So just look for your highest numbers and your lowest numbers and kind of roll with it. And if you're wondering the question that I'm referring to, AP Biology 2018 free response, I can post it so that you can take a look. I always think it's important to practice free response above all else. And the scoring guidelines are online too. So if you look at the first question in the link that I just posted, you will see the question that I am referring to from last year's AP exam. Okay. We're going to move away from evolution then. Any last questions before I move on? Speak now or forever hold your peace. All right. Let's keep cruising then. Okay, so I say picking back up because we did talk about this at one point. It was probably October, and a lot of you may not have been with us, but you can certainly go back and watch those casts because that'll be a whole hour about this stuff as opposed to 15 or 20 minutes. But we're going to talk a bit about cell structure. I'm going to highlight the major organelles that you should know. Vivek, great question. Yes, the G. gallus and A. forsteri can certainly be switched because they have this common rotation point. As well as kind of what I was saying, these two can be switched with these two. As long as E and E are paired and A and G are paired, right? It means the same thing because they can rotate around this axis. So you had a ton of freedom as long as A and G were together, E and E were together, and G was on its own. Okay? Cool. Okay, so I'm going to talk to you about the most important organelles that you need to be familiar with. If you have any quick questions, I can elaborate, but I'm more concerned with talking about the plasma membrane and cellular transport and then cellular respiration and photosynthesis. We may be able to do it all, which would be truly phenomenal. Okay, cell structure, organelles that you need to be familiar with. The mitochondria, which we're going to talk about a lot more when we talk about cellular respiration. If you've seen the memes, you know that the only thing that you learn in high school is that the mitochondria is the powerhouse of the cell. So of course you should know what the mitochondria does. It's going to produce almost all of our ATP. Very, very important organelle. Looks like that. Has the wavy lines in the inside, which is really important because we need lots of inner membrane in order to have our electron transport chain at its most efficient. So that is the mitochondria. The nucleus, also super important. It's obviously going to be protecting our DNA. It's also the difference between prokaryotes and eukaryotes. Prokaryote, karyote means like kernel, and pro means before. So it means before kernel, which means before nucleus. So that's why prokaryotes are called prokaryotes. Eu means with, so eukaryote with kernel, with nucleus. Here's your fun fact for the day, okay? But the nucleus, again, is going to be this membranous protection around DNA. Keeps it safe. It's going to lead to differences in how prokaryotes and eukaryotes transcribe and translate DNA. If you have questions about that, Next week, we'll be talking about all of those things. Endoplasmic reticulum, you need to know about the rough and the smooth endoplasmic reticulum. So the rough endoplasmic reticulum you can see here has all these little dots over it. It is called the rough endoplasmic reticulum because it's dotted with ribosomes, which I will show you on the next slide. That's where the majority of our proteins are going to be synthesized. And you'll notice it comes right after the nucleus, which is great. So mRNA comes right out the nucleus and gets translated into protein right in the rough endoplasmic reticulum. There's also the smooth endoplasmic reticulum, which is smooth because it lacks ribosomes. Therefore, it is not involved in protein synthesis. It is mainly involved in the synthesis of lipids and steroids. Lipids and steroids. Looks like we have a question. Sorry, this is a question about the table, so let me go back really quick. You need to read both the columns and the rows. You have to look in both locations, okay? So like I'll give you an example. So G. gallus, right? I'm looking here and I see 2 empty boxes. So we need to come back to those points, okay? And then I see that G. gallus has 3 differences with A. forsteri and 10 differences with E. africanus, okay? Where can I find the rest of the information about G. gallus's relationship with the other 2 organisms? I can find that in the G. gallus column. That's where I see that it has 11 differences with E. ferris and 18 with D. polylepis. Okay, so in order to read these, I'm going to need to read rows and I'm going to need to read columns at the same time. I find it way easier to just scan for highest and lowest numbers. So I'm scanning and I see a 1, so I know that E and E need to be paired together. I'm scanning and I see a 3, so I see that G and A need to be paired together. And that's going to be an easier way to navigate these tables. So hopefully that helped answer that question. Okay. All right. So those are your first organelles. Other organelles that you need to be familiar with, the Golgi apparatus. Love the Golgi. So this is involved, and there are some AP tests that I have looked over recently that were just super heavy on protein synthesis. Okay, and then you needed to know that protein synthesis is going to start with the ribosome. I mean, it starts in the nucleus, but that's not where protein is actually made. Okay, starts with the ribosome. That's where protein is first synthesized. Then it goes to the Golgi apparatus. We're going to come right after the rough endoplasmic reticulum. Looks like a stack of pancakes. And in the Golgi is where proteins are going to be processed. They're made pretty. We oftentimes turn them into glycoproteins, which is like a sugar protein. Think of glyco like glucose or glycolysis, right? So glycoproteins are going to be proteins that have sugar in them as well. And that's where all of that processing is going to take place. But again, they'll modify proteins, they'll trim them, they'll do whatever they need to do in the Golgi. So the streamline of proteins would be from rough endoplasmic reticulum to Golgi, and then usually to a vesicle. And you can see them right here, these little transport vesicles. They'll come and leave and deliver proteins where they need to be in the cell or outside of the cell. So a lot of signaling molecules get sent through vesicles. You also need to be familiar with the lysosome. Lys, L-Y-S, is going to be to break down. So if we talk about the cell lysing, that means that the cell is breaking or exploding. The lysosome has digestive enzymes in it, hydrolytic enzymes, things that are going to break other things down. And so they can help digest food, they can digest organelles, they can digest whole cells. They break down and recycle things for the cell if things are dead or broken. They do a lot of that kind of stuff, but they're really just breaking things down with those hydrolytic enzymes. The plasma membrane you want to be hyper familiar with because it's involved in all cellular transport, and I'm going to talk about this in more length in a second, but you need to know that we have a hydrophobic and hydrophilic region here based on where the phospholipid tails, or where the fatty acid tails and phosphorus heads are located, but I'm going to explain that more in a couple slides, so stay tuned. And then you need to be familiar with the chloroplast as well. We do not have a chloroplast. Plant cells have a chloroplast. That's how they make energy from the sun through photosynthesis. We'll talk more about that at the end if we have time when we talk about photosynthesis. Okay. Geometry dash, great question. Do we need to know anything about the nucleotide sequences that code for specific proteins, or are those usually laid out for us in problems? So I think you're talking about codon charts for translation. So that'll be a topic for next week. But the codon charts, you do not need to have any of those memorized. Like, you don't need to know that UGA is a stop codon. You don't need to know that AUG codes for methionine. That table would always be provided to you. You just need to be able to interpret it. So hopefully that helps answer that question for you. Okay, ooh, I included more. Wow, look at that. So vacuole is going to be storage. Vacuoles are really common in plant cells. Mainly the central water vacuole contains a lot of water for plant cells. They also have food vacuoles. The ribosome, which is where proteins are made. So you can see mRNA coming together with tRNA. That would be that codon chart where we find out what amino acid would be dropped off based on what the mRNA is coding for. That we're going to get into a lot more detail about next week, so stay tuned. And then vesicles I already showed you, but they're just little plasma membrane bubbles that are usually bringing things out of the cell or into the cell or moving things around the cell. I always think vesicle, vehicle, so they're the vehicles of the cell. David, you get a link to the slides if you are a Fiveable Plus member, which you can sign up for on our website. Okay. So just brief review of prokaryotes versus eukaryotes. And some of this we'll talk about next week as well with transcription and translation. So we already mentioned the fact that we have no nucleus versus nucleus for prokaryotes and eukaryotes. Important to know that prokaryotes do have cell walls, cell membranes, ribosomes, and DNA. Cell walls, cell membranes, ribosomes, and DNA. They have a single circular piece of DNA, which differentiates them. And their ribosomes are not connected to an organelle like ours are, but they do have all of those things. So common misconception is that people will always write a main difference between prokaryotes and eukaryotes is that one has a cell wall and one doesn't. There's a lot of things wrong with that answer. One is that you're saying one has a cell wall and one doesn't. Even if you know which one has a cell wall, your reader doesn't know that you know that, so you get 0 points. So you always need to be specific, okay? Or if you just said, prokaryotes have a cell wall, period, right? You are referring then that eukaryotes do not, which is not true, first of all, but that's besides the point. But they still won't give you a point for that because you didn't make it explicitly clear what the role of the eukaryotes was in that situation. You always want to write both sides of the coin there, right? If you're trying to compare and contrast, you need to mention what's occurring in both groups. Otherwise, you get no points. I have had students cry over this before. It is legitimate. It is for real. Make sure that you follow those instructions. The other major problem is that that answer is not even correct because plants have cell walls and plants are eukaryotic cells. So students oftentimes forget, they remember that prokaryotes have cell walls and then forget that plant cells are eukaryotic and therefore blah, 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 blah. It's not a correct answer. Also can't be so vague like that in your answers. It's got to be really, really specific when you're writing these free responses. And that's why I think it's really important to continually look at rubrics of how free responses are graded so that you know exactly what AP is expecting of you. You can try to live up to those expectations because they're very challenging. Okay, so yeah, important to remember that prokaryotes do have ribosomes. They do make ATP. They just go through glycolysis. So that's another one that students sometimes forget. But all living things, in order to be considered living, need to have their own metabolism and make their own energy. Other things, so I have only go through glycolysis on there. They do not have any organelles, no membrane-bound organelles, that is. And they have transcription and translation happening at the same time because they don't have a nucleus, right? And again, I'm going to touch more on that next week. So if you have questions about that, kind of hold off on that one. Eukaryotes obviously have a nucleus. They have multiple linear strands of DNA. They have histones in their DNA, which is just like a little bead that we wrap our DNA around. Kind of think of like a spool of thread. As opposed to having 50 yards of thread, we have like a ½ an inch of thread that's wrapped around a spool. We have lots of different membrane-bound organelles. We have transcription and translation happen separately. All sorts of good things. Plants have cell walls. All cells have a cell membrane. What kind of organisms are prokaryotes? Great question. Bacteria are going to be the main prokaryotes. So anything that's in the archaea or bacteria kingdoms. So we have eukarya, archaea, and bacteria as our three, not kingdoms, domains. Kingdoms is old speak. Those don't really exist anymore. But those are our main divisions. So any kind of archaea or bacteria are going to be prokaryotic. Eukaryotes are going to be pretty much everything else that you can think of. Usually multi-celled organisms. Yeast are single-celled eukaryotes, almost everything else is going to be a multicellular organism. Prokaryotes are all single cellular organisms. There's no multicellular prokaryotes. Okay, so those are important distinguishing factors. Now let's talk a little bit more about that plasma membrane and how it's selectively permeable. So I said we'd dive a little bit deeper into this part of cells. So remember that the plasma membrane is not just on the outside of the cell, but also around all of the organelles, which is why we call them membrane-bound organelles in a eukaryote. Also makes up the nucleus in a eukaryote and the little vesicles that I was showing you. So it's a very, very important structure, and it has everything to do with what can and cannot get into a cell, which you do need to be highly familiar with. So what you can see here is that our plasma membrane is made up of what we call a phospholipid bilayer. So let me type that into the box so that you can see what I'm talking about. Phospholipid bilayer. Okay, so I'll break that word down for you. So phospholipid means we have a phosphate and we have a lipid, right? Phospholipid. And what that looks like here is this phosphorus head group and then this lipid tail group, which is going to be really nonpolar and hydrophobic, right? Because lipids are fats. Fat and water do not mix. Oil and water do not mix, right? And then bilayer is because we have two layers here, okay? And that helps to create this kind of selectively permeable membrane, because you'll notice that they're going in the reverse direction, so that our phosphate groups, which are hydrophilic, meaning water-loving, are on the outside or inside of the cell where water is present. Hey, water, water. And then our hydrophobic or water-hating, afraid of water, tails, those fats, are going to be protected on the inside of this bilayer. Okay, so they're oriented like this. These tails are going to prevent anything that is polar or water-like or charged from getting through the membrane. And then the heads, which are totally chill with water, are going to kind of keep the inside tails protected. So here you can see the water molecules are interacting with the phosphate heads, but no water molecules are interacting with these fatty acid, very non-polar tails. And I have a whole thing on biochemistry that you can rewatch if you're unfamiliar with some of the terminology that I'm using. But in brief, polarity is really referring to how charge is shared in a molecule. So water is a polar molecule because it has an unequal distribution of charge. Oxygen is very electronegative, so therefore it's pulling a lot of negativity towards the oxygen. And then we have those two hydrogen molecules, which are positively charged. So we have this unequal distribution of charge with a negative oxygen and positive hydrogens. It all cancels each other out, and we have a net neutral molecule. There are partial charges, is what they're called, within the molecule. And if you've taken chemistry, I'm sure you're familiar with that. Nonpolar molecules are going to be completely equally shared, evenly distributed charges, and they are going to not dissociate in water. So an example, salt would be polar, sugar would be nonpolar. If you put sugar in water, it's going to say as C6H12O6. It's going to break down into little structures of itself, but it's not going to break apart. Whereas salt, NaCl, if you put that in water, it will dissociate into Na plus and Cl minus. We have those partial charges that are now manifesting. That bond would break in water, and those kinds of things that have a charge on them would not be capable of getting through the plasma membrane on their own, so instead we would have to use a transport protein. The same is true for water. Water cannot get through the plasma membrane on its own because of those fatty acid water-hating tails. Again, think oil and water don't mix. These are fatty tails. Water is going to be repelled by those. Okay, and so therefore water is not going to be able to get through the membrane without the help of a transport protein, which I will show you in a second. Oops. Okay, there's also proteins, lots of different proteins embedded in the plasma membrane. We call it the fluid mosaic model. It's kind of just this like ever-moving kind of sea of molecules. And you can see these proteins are embedded in it, and they're going to have various functions. Some of them act as enzymes, some of them act as transport proteins, but they have hydrophobic and hydrophilic regions as well. And you can see those there. They're going to correspond to whether or not that part of the protein is interacting with water. Okay, so in order to then get things happening here, our plasma membrane has a lot of different roles. And we can see a few of them here. So transport is going to be a big one. We need to get things into and out of the cell, and we can do so passively or actively, which I will allude to a little bit more in a second. They can also act as enzymes, which are going to lead to enzymatic activity within the cell. Okay, so enzymes are always going to be proteins that help to speed up a reaction. Signaling molecules. So oftentimes a signaling molecule will bind to a very specific receptor in the plasma membrane and therefore cause a reaction or a signal transduction. Cell-to-cell recognition. This is very, very common, especially with the immune system, when we have like a T cell recognizing an antigen on a macrophage or something like that. And we'll review that in a couple of weeks. When we have that recognition, that's all happening through the plasma membrane, cells joining one another, and then attachment to the cytoskeleton if the cell is moving. Sorry, again, I feel like I'm losing my voice. Review season is upon us, and I'm talking so much in class. It's all good stuff. So this is just an overview of all the different things that the plasma membrane helps contribute to. We're going to talk today mainly about transport, because that's going to be one of the most important ones. So when we're talking about transport, you need to be familiar with this terminology called the concentration gradient. And a gradient is an incline, a hill, a non-flat line. And so you may have seen like a road sign if you've ever driven through mountains, and they'll be like, the gradient is 20, and it means it's like a 20% incline. And they'll tell you, because trucks especially need to know how steep the hill is, but that's all a gradient is a like a degree above the horizon, I suppose we could call it. So a concentration gradient is going to be an uneven distribution of molecules. So what we can see here is that we have a concentration of molecules on the left and a concentration of molecules on the right. If you picture this as an actual gradient, when you have a high concentration, whoop, we're all the way up here. When you have a low concentration, whoop, you're all the way down here. And molecules are going to flow down a gradient. They're going to flow downhill, right, from high to low. I always explain this to my students as like human nature. Humans want to spread out. If I tried to shove all 1,200 students that go to my school inside of my classroom, I would have to expend a lot of energy to do that, because I'm taking all these students who are all spread out and I'm cramming them into a highly concentrated room, right? But if I cram all 1,200 students into my classroom and then I open the door and I let them leave, they will do so willingly, and they will probably stampede me, and I will no longer exist as a human anymore from shoving them all in my room. But very, very high concentration, they naturally want to spread out. Humans naturally want to spread out. Molecules naturally want to spread out. And that's just like basic kinetics too. So if you've talked in physics or chemistry about the movement of molecules, they bounce against one another and therefore repel one another. So in a highly concentrated area, if there's more space elsewhere, they will bounce each other into that more spread out area and therefore into a lower concentration. Okay, so with this, we have a lot of different applications. So there's diffusion, which is when we have some sort of barrier. Okay, so we have a high concentration on the left and a low concentration on the right. And over time, the goal is always equilibrium, right? So while at first we're going to have a net movement of molecules over to the right, eventually we will have no net movement. Maybe molecules will slowly be bouncing back and forth between the sides, but we're going to have an equal distribution of molecules on both sides here so that we're perfectly spread out, and that would be considered equilibrium. So that's what's going to happen over time, but the net movement of molecules would be to the right. So within this kind of umbrella, we can talk about passive transport. So with diffusion and facilitated diffusion, all of this type of movement, and you do need to be familiar with this vocabulary, is passive, right? And passive means without energy, right? If you are a very passive person, you're not putting up a fight. You're not expending energy in order to prove a point, okay? So passive transport does not require energy. This means that molecules are going to naturally be moving then from a high concentration to a low concentration, which is how molecules like to move because they like to be spread out. On the left-hand side, we have your classic diffusion. These molecules are able to pass through the membrane. They are uncharged, nonpolar molecules. Carbon dioxide and oxygen are great examples of this. They flow freely into or out of the cell depending on where there's a higher or lower concentration, which is exactly what we're seeing with these purple molecules on the left, moving from high to low. On the right-hand side, we have facilitated diffusion. Still diffusion, still passive transport, still not requiring energy, but we need a facilitator now. We need something to assist, and that's why we have these protein channels. So we have a channel protein and a carrier protein. You can see the carrier protein can open and close, and the channel just stays open. And this is going to allow polar, hydrophilic, water-loving, polar, maybe charged molecules through the membrane. So like the sodium-potassium channels when we talk about action potential in the nervous system. Sodium and potassium have to travel through a protein channel because sodium is Na+, potassium is K+. Both of those are charged, and therefore they won't be able to make it through the membrane without the help of a protein protecting them, because as you can see, they do not need to interact now with those fatty acid hydrophobic tails if they have this little channel or this little bead through the membrane to protect them. Okay, so all of these things require zero energy, and all of these molecules are moving from high to low. The only difference is whether or not they need the help of a protein to get through the membrane. Another type of transport that's also passive, meaning that it does not require energy, is osmosis. And osmosis is just a fancy word for the movement of water molecules. Okay, so the only thing that sometimes confuses students here, because osmosis is super important, is the way that water moves can be kind of, it's not counterintuitive. Water is still moving from high to low, just like every other substance on Earth. But the difference is where water is lower concentrated. So if this was, I should have included a better diagram. If these water levels were equal, right, and we had all of this solute or stuff, solute is just dissolved substance, over on the right-hand side, then there's less water on the right-hand side because there's more other stuff there. And if you have other stuff in your water, then you have less water, right? So if we have like 10% salt over here, then this solution is 90% water, as opposed to over here where maybe we only have 1% salt, and therefore we have 99% water. So water is going to move to where there's more solute or more stuff, because that's where there's less water molecules. Again, for the sole reason that there's other things there. So we can see water does indeed move through this semi-permeable membrane, and that's why we have an increased amount of water on the right side in comparison to the left side. We actually would have equal amounts of water molecules on either side. It just looks like there's more water on the right-hand side because there's other stuff that's taking up room there that's not water molecules, and therefore it looks like there's more stuff over on the right. Okay. Other types of transport to be familiar with. So facilitated diffusion I already talked about, but there's your difference channel and carrier protein. And then lastly, active transport. So active is going to be the antithesis of passive, where passive does not require energy, active does require energy. Just like if you are an active person, you're requiring energy, you're using energy to be active. Right? And so usually 9 times out of 10, when we're using active transport, it's because we're moving molecules from a low concentration to a high concentration where they don't want to go. So we'll see this in a couple different instances. We actually move things actively across the plasma membrane in cellular respiration if we have time to talk about it, which it doesn't look like we will. So that's a good place to start next week. But this also happens with the sodium-potassium pump when we're trying to create a concentration gradient inside of the neuron. So we'll pump sodium outside of the cell, will pump potassium inside of the cell, even though neither of those things want to go there because it's going against the concentration gradient. Okay, so those things are active, they require ATP, and they're relatively common. Again, it's happening in your brain all the time. All your neurons are using active transport all the time, and ATP in many different ways. Okay. So going along, and then we'll kind of wrap up. Let me just see how many more slides I've been in here. We can like very briefly start cellular respiration, or I can just take a few questions for the last few minutes. But we'll wrap up kind of this idea of cellular transport by talking about 3 other vocabulary words that you need to be familiar with, and that is hyper, iso, and hypotonic. And so those terminologies you need to be careful with because you need to know if it's referring to the environment or the cell. And in this case, usually it's talking about the solution that a cell is in. Is the solution hypertonic, isotonic, or hypotonic? It's always a comparison. So for instance, hypertonic just means that there's more solutes, there's more other stuff outside of the cell than there is inside of the cell. So if we injected your body with really salty water, like it was 25% saline, and our body is only about 1% saline or 1% salt. If we injected your body with 25% salt, water would start to leave your cells and go into the highly concentrated outer environment. Okay, so water is leaving. Hypertonic, it's going to where there's more solute, more stuff, and you can see these cells are shriveling up. Okay, I always remind my students to remember this, because if you're hyper, you run outside, right? So if you're hypertonic, water is running out, meaning there's more stuff outside of the cell than inside of the cell. In the middle, you say isotonic, and iso is going to be equal. That means there's the same amount of stuff inside of the cell as outside of the cell. That doesn't mean there's no stuff inside of the cell. As I said, our bodies are 1% saline. I think I'm quoting that right, 0.1%, 0.1 or 1, don't quote me. Regardless, so when you get an IV, it's not pure water, right? When you get an IV, it's actually 1% or 0.1% or whatever it is, saline or salt solution, that's what that means. And so your cells are really, really happy because there's the same amount of stuff inside of them as there is outside of them, and therefore water is moving freely inside and outside, but it's not being drawn one way or the other. Lastly, hypotonic is going to be as if we injected pure water into your body, meaning that there's more stuff inside of the cell than outside of the cell. Therefore, water is going to pour inside of the cell where there's less of it. So it's imploding into the cell where there's more solute and less water, and those cells will eventually explode. So if your IVs were plain water, it would not end well. Okay, you'd have a lot of lysed or popped red blood cells. And so I always tell my students, remember this one, hypo is like hippo, hypo. If you're a lazy hippo, you go inside, right? And you go sit on the couch and be a couch potato. So hyper, you run outside. Hippo, you go inside, which means water is moving inside of the cell. And then iso is just going to be equal. Okay, Vivek, I can answer your question briefly, and then we'll talk about it more when we talk about the nervous system, which should be in 2 weeks. But your neurons use active transport all the time because they have to create a concentration gradient in order for an action potential to occur. So an action potential all happens with depolarizing our membranes. And again, it's going to be hard for me to explain this simply if you're not already familiar. But our neurons are negatively charged. And that's because there's a lot more positive charge outside. We pump a lot of sodium out of the cell, and there's a lot less potassium inside, and there's also a lot of negative charges inside of the cell. So when an action potential occurs, like we have a thought or our neurons get a signal, we open up sodium channels, and sodium comes rushing in and repolarizes the membrane, making it nice and positive. But we need to eventually reset that and pump that sodium back out so that we can do it all over again. It's kind of like you knock down the dominoes and then you need to set all the dominoes back up. And the setting of the dominoes back up, the resetting of our neuron once we have an action potential, is what active transport is used for. And that's all has to do with the sodium-potassium pump. And if you have a more pressing question that needs to be answered before we talk about it in detail, and I can show you lots of diagrams because it's always hard for me to kind of just like verbally explain without showing you. I'm a very visual learner. I would just suggest that you look up the sodium-potassium pump because that's going to be active transport. Oh, good. I'm so glad that helps. Any other closing questions before we wrap up? We have a few minutes left if anyone has any final questions. And I will for sure talk about cellular respiration and photosynthesis at the top of next week's episode. And then we'll jump into DNA and transcription and translation and genetics. It'll be a big week. We'll cover a lot of topics. So definitely stay tuned. Any other questions though? Vivek, that is a great question. I'm sorry if I'm mispronouncing your name. But so we'll have one on the 17th. We'll have one, so that's next week, even though it's spring break, it's still going to be a really big week. So definitely tune in. We'll have one on the 24th. We'll have one on the 1st. We'll have one on the 8th. And I think we might also have on the 9th, 10th, and 11th. So it's actually 1, 2, 3, 4. We'll definitely have 4 sessions for everyone. And then we'll probably have 3 additional cram sessions. before the AP exam, like the weekend before. Trevor, also a great question. So I have fully covered ecology in a couple podcasts that have already happened, but we haven't reviewed it yet. So last week was our first week of review, and we just talked about evolution. And then you were obviously with us this week when we talked about evolution and cells. So I haven't fully reviewed ecology yet, but I have gone through it all the first time around, which would be the longest time that I'll talk about it, because with review I just don't have as much time. So if you watch the replays, I did that pretty recently within the last month. The slides are not posted in here anymore. The slides are only posted if you are a Fiveable Plus member. Then they're all posted on the website, and you can access them all there. Any other questions? I would highly suggest a Fiveable Plus membership also, just because it is, you know, I think it's $25. It may be up to $35 now. Shoot, I should really know that. But it's an investment, and you get a lot of individualized review closer to the AP exam and access to all my slides and replays so that you can watch me talking. It does cost for Fiveable Plus. I can't believe that I don't know this. I think we just upped it to $35. But let me really quickly hop on the website and just make sure that I'm not saying something wrong. And then I'll get you an answer. But then you'll have access to the entire library of videos since September, so you can rewatch videos in detail that you need help with. And then you don't have to, or you'll get more information on those topics. Okay, so Fiveable Plus is $35. So it's $35. You get access to all replays. You get trivia questions on online, and we have an app coming out. You get extra support, extra cram sessions, all sorts of fun things. But unlimited replays, archives of notes, slides, study materials, all of those types of things are available to you for $35. I should have posted our website when I just did that. That was dumb of me. I'll put that up there right now so that you can hop in and subscribe. And again, I have a whole library of hour-long videos that have been playing since September. So if there's specific topics that you need extra help with, yeah, it's $35 for the whole year. So it's a pretty good deal, and you'll have access to all of my content. You also have access to all other, if you're in other AP subjects, you can have access to all of those videos as well. All right, so the link is there. If you think of any questions, bring them with you next week. I would love to answer them and make sure that you're getting all the individualized support that you need. Sign up for a membership. It's a really great opportunity. It's a good investment in you passing the AP exam, which is, of course, the goal, getting some college credit and all that good stuff. So come with your questions. I'll be ready for you. Be happy to answer anything. Thanks for participating tonight. This was a great group. Let your friends know what we're talking about next week, because it's a big one. And I look forward to seeing you. I hope you have a restful spring break and have some really good quality study time, but also rest time, because rest is equally as important. All right, y'all. Hopefully I see you all next week. Yeah, have a great week. See you then.